So triacylglycerols are basically esters of fatty acids and the molecule glycerol. So if we look at our structure over here, this portion of the molecule comes from glycerol. It's basically a 3-carbon molecule with 3 alcohol functionalities on it. And we form 3 esters between glycerol and fatty acids, and that gives rise to the triacylglycerols. In general, there are 2 classes of fats that come out of this. First, we have fats, which are generally solid at room temperature. They are that way because they contain a fairly large amount of saturated fatty acids. So good example of this, butter or lard. These are our typical fats. Oils, on the other hand, tend to be liquid at room temperature. And they tend to contain a fairly large amount of unsaturated fatty acids. Basically, the packing that we saw between the fatty acids carries over to the packing that is seen in triacylglycerols. And in general, fats are going to have a lot of close packing, whereas oils will have the packing relatively disrupted. Now, in terms of the functions of triacylglycerols, really one of the big things is energy storage. So in animals, we have adipocytes, adipose tissue. Plants store fatty acids as well. In seeds, basically fatty acids are going to be the energy for developing seeds until the process of photosynthesis can kick in and the plant can start making energy from the sun. We also use them as a source of insulation and water repellent nature. Basically, a lot of marine animals tend to have a pretty thick layer of triacylglycerol somewhere on their body, and basically this is meant simply to prevent the loss of heat. So animals that live in very cold environments, they tend to have a fairly large amount of triacylglycerols to prevent heat loss. Last thing here, soap. The initial way we made soap was by taking triacylglycerols and treating it with sodium hydroxide. And this basically gave a large number of fatty acids that served very nicely for making micelles and basically allowed us to effectively wash our hands. So we had a fairly good method for basically washing our hands thanks to the lipids that we found in various organisms. So phospholipids are a component of membranes. They're also used in our food as emulsification agents. So what we see in a phospholipid, we still have a molecule of glycerol right here, and we attach 2 fatty acids to that, and then the third site is taken up by a molecule of phosphoric acid. It's basically a phosphate. So and that's why we call this a phospholipid. Now, in terms of phospholipids, they line up very nicely with other phospholipids, and they tend to make very nice membrane structures. The phospholipid bilayer basically arises from this. Now, a lot of times with phosphates, we tend to attach other things to this phosphate. And that actually differentiates the different phosphates. In fact, generally in any sort of lipid bilayer, we see an inequality in the distribution of the different phospholipids across the membrane. So when we look at a phospholipid bilayer, basically this is the phosphate head right here. Phosphate head is right here. Here are the 2 hydrocarbon tails from the fatty acids that are attached, and they line up very nicely with each other, you'll notice here. And when they do so, basically the ends of the hydrocarbon tails match up very nicely with another set of ends of hydrocarbon tails, and that's how we end up forming this bilayer. So polar head up here, non-polar tail down here, basically highly hydrophobic region in here, and region here that wants to associate with an aqueous environment. That's going to lead very nicely to the formation of a membrane structure. Now, as I mentioned, we can attach a variety of other things to phospholipids. So, for example, the amino acid serine very often gets attached to phospholipids. As we see here, this is basically serine right there, and it gives rise to a molecule called phosphatidylserine. Same thing with choline. This molecule right here is choline. This gives rise to phosphatidylcholine, which is also known as lecithin. If you ever look on at the ingredients in a milk chocolate bar, most likely you will see lecithin. That's essentially what lecithin is phosphatidylcholine. Phosphatidylcholine and phosphatidylethynolamine are very similar in structure. Choline just has basically is phosphatidylethynolamine with 3 methyl groups on the amine group there. Another example takes a sugar derivative. This is inositol right there, and sticks it on the phosphate, and that gives rise to phosphatidylinositol. And then finally, if we take 2 phospholipids and basically link them together using a molecule of glycerol, this gives rise to a molecule called diphosphatidylglycerol, also known as cardiolipin. And this is found in very high amounts in our heart tissue. So as a result, basically fairly large structure in comparison to other phospholipids, basically because you're joining 2 phospholipids together. Now, sphingolipids have some similar properties to phospholipids. Basically, we start off with a molecule of sphingosine, which arises, this portion here arises from palmitate, this portion here from serine. Now, sphingosine has a fatty acid attached to it. The fatty acid actually gets attached to the amine group that came from serine, and this gives rise to a molecule that we call a ceramide. Ceramides then get further modified, first by adding a phosphate, and then by adding an alcohol, kind of like we saw in the phospholipids. So in this case, if we add a molecule of choline, we end up making a molecule called sphingomyelin. Sphingomyelin is found in relatively high concentrations in the nerve cells, particularly in the membranes of our nerve cells. Basically, it's found in other cells as well, but really the highest concentration is found in the membrane of nerve cells. So sphingolipids are used in pretty much all membranes, but obviously certain of them end up enriched in certain membranes. Now, we can do one other thing. We can actually take a carbohydrate and attach the carbohydrate to ceramide. And what that does is gives rise to what are called cerebrosides and gangliosides, which also tend to end up in nervous tissue as well, in fairly high content. And they are relatively interesting in terms of the fact that they're simply, predominantly a lipid, but they do have a sugar attached to them.